Welcome to another video. Today our topic is Mendel's law of segregation. So, this fundamental principles of inheritance was discovered by Gregor Mendel through its experiments with pea plants. Let's dive into the key concept, experiments and implications of this law. Mendel's law of segregation explains how alleles, which are different versions of a gene, separate during the formation of gametes. The reproductive cells, such as sperms and eggs. This law forms the fundamental of classical genetics, helping us understand how traits are passed from one generation to the next. To understand Mendel's law, we need to familiarize ourselves with a few key concepts. Alleles. These are different versions of a gene. For example, the gene for seed shape in pea plant has two alleles, one for the round seed and one for the wrinkled seed. Homozygous, an organism with two identical alleles for a trait such as double R capital or double R small. Heterozygous, an organism with two different alleles for a trait such as capital R with small r. Genotype, the genetic makeup of an organism represented by its allele like capital double R or capital R with a small r or small double R. Phenotype, the observable characteristics or traits such as round or wrinkled seeds are known as phenotype. Mendel's experiments. Mendel conducted his experiments using pea plants and focused on monohybrid crosses, where he crossed two breeding plants with different traits. For instance, he crossed plants with round seeds with plants with the wrinkled seeds of the first generation or F1 generation consisted entirely of heterozygous plants which was the capital R with a small r, all displaying the dominant round seed trait. He then allowed the F1 generation to self-pollinate, resulting in the second generation, or F2. The F2 generation showed a 3:1 phenotype ratio of round to wrinkled seeds, revealing the segregation of alleles. From these experiments, Mendel proposed the law of segregation, which has several key points. 1. Each individual has two alleles for each trait, one inherited from each parent. During gametes formation, these alleles segregate so that each gamete carries only one allele for each trait. During fertilization, the fusion of male and female gametes is random, restoring the diploid state in the offspring and determining their genotype. This segregation ensures genetic diversity in the offspring. Dominant and recessive characters. In Mendel's experiment, he discovered the concept of dominant and recessive characters. A dominant character is an allele that expresses its phenotype even when only one copy is present. For example, the allele for round seed, which is the capital R, is dominant, so plants with the genotype double capital R or capital R with a small r will have round seeds. A recessive character is an allele that only expresses its phenotype when two copies are present. For example, the alleles for wrinkled seed like a small r is recessive, so plants must have the genotype double small r to display its wrinkled seeds. So, Mendel's law of segregation is based on several assumptions. One, the trait is controlled by a single gene with two alleles. The one allele is dominant and the other is recessive. Third one is the combination of allele in the offspring occurs randomly during fertilization. Fourth one is each pair of allele segregates independently of other pairs providing the genes are not linked. To visualize how alleles segregate and combine, we use a tool called a Punnett square. Let's set up a Punnett square for a monohybrid cross of two heterozygous plants. First, draw a 2 into 2 grid. Next, label of row and column with the allele of each parent. For example, place capital R and small r across the top for one parent and a small and a large R down the side for the other parent. Now fill in the sequence by combining the allele from the top row and side column. This result in four possible combinations: the double capital R with a small r, capital R with a small r, and small double R. Each square represents a possible genotype of the offspring. So the genotypic ratio is 1 for the double R, which is the capital one, and 2 ratio 2 is for the capital R with a small r, and 1 for the small double R. This means there are 3 round seed plants for every one wrinkled seed plant, giving us a phenotypic ratio of 3 ratio 1. Genotypic and phenotypic ratio. Summarizing the result for the Punnett square, genotypic ratio is 1 ratio 2 ratio 1. For the phenotypic ratio, this is the 3 for the round seed and 1 for the wrinkled seed. These ratios help us predict the distribution of inherited trait in offspring. So, in the conclusion, Mendel's law of segregation explains how alleles segregate during gamete formation and how they combine during fertilization. Dominant and recessive allele determine phenotype expression, and Punnett square are useful tools for predicting the genetic outcomes. This law forms the basis for understanding genetic inheritance pattern and is crucial for the study of genetics. Please do like, share, and subscribe our channel. Thank you.